Welcome children. In this video I will be dealing with the topic mutations. It is coming from two chapters, chapter 5 and chapter 6, both belonging to genetics. What is mutation? Mutation refers to alterations in the DNA sequence resulting in changes in the genotype and hence the phenotype of an organism. So, we have the DNA with a specific sequence. If it gets altered, it will result in changes in the genotype, and if the changes are very drastic, it will be shown in the phenotype. We know many of the cancers are caused due to mutations in the DNA. Now, let us analyze those agents which induces mutation. Those agents are called mutagens. So, agents that induces mutation are called mutagens. They include physical mutagens, chemical mutagens and biological mutagens. Some physical mutagens include gamma rays, beta rays, X rays, the UVB rays of the sun. These are some examples of physical mutagens. Chemical mutagens, we know nicotine present in cigarettes, then certain pesticides like endosulfan, etc. are mutagenic, that is they cause mutations or cause chromosomal or DNA alterations, and biological mutations are caused due to biological agents like oncogenic viruses. They also brings about mutation. So, this slide deals with the topic what is mutation and the mutation causing agents called mutagen, which are broadly classified into physical, chemical and biological agents. Types of mutations. Basically, there are two types. They are gene mutations and chromosomal mutations. Gene mutation is at the gene level and chromosomal mutation is at the chromosomal level. You know what is a gene? Basic structure of the or segments of the DNA. So, at that level it is called gene mutation, and chromosomes, we know the DNA is packed to form the chromosomes, and we have seen chromosomes at different stages of cell division with respect to prophase or metaphase, anaphase. You have an idea of chromosomes, and we know chromosome is a packaging of the DNA and how the DNA is packaged into the chromosome with the help of histones and non-histone chromosome proteins. So, when mutation occurs at the gene level, it is called gene mutation, and when mutation occurs at the chromosomal level, it is called chromosomal mutation. So, these are broadly two types, gene mutation and chromosomal mutation. Now, gene mutations are of two types, point mutations and frame shift mutation. Point mutation, you have learned the example of the disease sickle cell anemia. It is caused due to the change in a single base pair at a point. So, I will go to the next slide to explain that. Let this be the DNA. Now, I am unwinding the DNA to specify the mutation. We know that at a particular point mutation has taken place in sickle cell anemia. It is supposed to be GAG, the normal coding strand. We know the two strands by now. We know the two strands of the DNA are coding strand and template strand, and we know the coding strand gives us the idea of the mRNA. The mRNA which is running in the 5' 3' direction will be exact base copies of the coding strand provided that T is replaced by U. So, if this is the sequence, I am only pinpointing at the where mutation is occurring. GAG is the coding is a sequence in the coding strand. So, automatically what will be the sequence corresponding on the template strand? For G it is C, for A it is T and G it is C. And on the template strand only the mRNA is synthesized, which will be complementary to C, that is G, T it is A and C it is G, which means that we know that mRNA is equal to coding strand with respect to the sequences of bases. Now, and which is the amino acid that comes to GAG. Remember, it is a glutamic acid. So this is the normal beta globin chain. There are a number of codons here which are correct, there are a number, but I am talking only of the 6th position. Here it is a 6th codon. So this will be the 6th amino acid in the proteins. So this is normal. GAG is normal, there is no mutation taking place. Therefore, the mRNA that is transcribed will be GAG, and the proteins that is translated also will be a normal beta globin chain with the 6th position having glutamic acid, which is normal. But now we are going to introduce mutation here. With another color I am going to show mutation has taken place. It is occurring at a single point or at a single base pair. What is gene mutation? Gene mutation or point mutation is the change in a single base pair. Change in a single base pair. Highlighted, single base pair. So now here what happens, instead of A, it becomes T. So due to mutation, may be physical or chemical or biological, instead of A, which is normal, it became T. So correspondingly, what will be the mRNA synthesized? It will be GUG. If it's GUG, what will be the amino acid? Will it be glutamic acid that is coming to the 6th position? No, it will be a wrong amino acid called valine. So what happens in the 6th position of the beta globin chain? A wrong amino acid binds, that is valine instead of glutamic acid. And this alteration results in sickle cell anemia, where the RBC gets a distorted shape, the sickle shape, not a disc shape. So you understood where the mutation has taken place, at a single base pair or at a point. Instead of GUG, it becomes GTG, means T. Instead of U, it is replaced by T. It's a single base change. There is a change. Instead of U, it became T, and correspondingly what happens, the mRNA synthesized also will be changed. It is GUG, which means not glutamic acid that is coming in the 6th position of the beta globin chain. It's a wrong amino acid called valine. And that's how the person gets sickle cell anemia, a condition where oxygen carrying capacity of the person is lost. So this is a very good example of point mutation. Next, frame shift mutation. We have learned this, which is on page 1113. We learned it with an example or an exercise. Ram has red cap. So we took such a sentence because we know that codons are a triplet basis. So now what we have analyzed that time when we did this exercise, when one or two bases are deleted or added. See, I am going to delete. I am going to delete M and I am going to delete A. So here 1 or 2. Remember 1 or 2, what happens? The reading frame gets altered. So now we have to reshuffle. This will join RHA, SRE, DCA and P. Even if it's 1 also, this a alteration or the reading frame gets altered. But on the other hand, again Ram has red cap. If 3 bases are removed or multiples of 3, 6 are removed, is the reading frame altered from the point of mutation? See, from this point, can you read? Yes, we can read it as cap. Okay. So remember, in frame shift mutation, you give the clause when. So you start it with a clause when 3 or multiples of 3 bases are deleted or inserted. We can insert or delete. The reading frame remains unaltered. This is called frame shift mutation. So, I hope you have understood frame shift mutation with the exercise RAM has red cap, which we have already done, which is on page 113. There we notice if one or two bases are deleted or inserted. In both cases, the reading frame is altered. Whereas, if it is 3 or multiples of 3, the reading frame does not get altered from the point of mutation or from the point of insertion or from the point of deletion. So, I hope you have understood gene mutation, which is occurring at the gene level. We have point mutation. To explain, we have the sickle cell anemia, and frame shift mutation, we have the exercise RAM has red cap. That also serves as an evidence that codons appear as triplet bases. Now, moving on to chromosomal mutations. Chromosomal mutations are divided into two types. They are 1 structural mutation, 2 numerical mutation. Structural mutation, I will give it with an illustration. Consider this to be a chromosome with segments A, B, C, D, E, F, G, H. So, let this be a normal chromosome with these genes. Here gene stands for segments and each gene is represented by an alphabet, namely A, B, C, D, E, F, G, H. So, you know the length of the normal gene is or DNA is so much, correct length. But we are going to see a situation where mutation has taken place, either deletion or addition of a segment. We are going to take the case of BC getting deleted in the first case. So, we are going to get it A, D, E, F, G, H. Now, look at the DNA structure. It has been altered. It was so lengthy, but now it has got reduced in structure. The reduction in structure is because of the deletion of the segment BC. So, we see this condition is called as deletion. Now, opposite of this condition is duplication, that is gain of a segment. So, again let it be BC. Here the mutation is occurring at BC. Now, here BC is becoming duplicated, BC, BC, then D, E, F, G, H. Now, you see what has happened to the length of the chromosome from the normal again deviated, it became more lengthy. So, this is also a structural aberration or structural mutation where it has altered from the normal structure by addition or by gaining a segment. The segment gained is BC, see BC. So, in these two cases, deletion and duplication, the structure of the chromosome gets altered either by losing a segment or by gaining a segment. Clear? So, under structural mutation, we have two types. They are deletions and duplications. In both the cases, the structure is getting deviated from the normal size. In the first case, the structure of the chromosome becomes smaller. Second case, it becomes longer than the normal. And the first case is due to the loss of a segment. In the second case is due to the gain of a segment of the chromosome. It can be that segment is indicating a gene segment or a portion of the chromosome. So, I hope you have understood structural mutation. Next, moving on to numerical mutation. Numerical mutations can be represented, or it is divided into two. They are aneuploidy and polyploidy. And again, aneuploidy is further divided into two. They are, with this representation you will understand, 2n minus 1 means monosomy and 2n plus 1 is trisomy. And polyploidy can be represented, the normal ploidy is 2n. So, it is not 2n, it is becoming 3n, 4n, 5n, 6n, etc., etc. So, once again concentrate at numerical aberrations or numerical mutation. It is a change in the number. Now, to talk about aneuploidy, it is a change in the number of chromosomes. Once again, aneuploidy is a change in the number of chromosomes. If you analyze this, how is the change in the number of chromosome? Here the change in the number of chromosome due to the loss of one chromosome. So if the human chromosome 23 into 2, it is 46, but if it is going to undergo monosomy, it will be 45. So here we can say change in the number of chromosome due to the loss of a chromosome. In that case, it is represented by mathematical form 2n minus 1. That is called monosomy. Now the very similar situation, we know 23 into 2 is 46, but sometimes it can become 47. How it became 47? That is due to the addition or due to the gain of one chromosome. Then it is called trisomy. So here in aneuploidy, remember the frame of the sentence, change in the number of chromosome either by the loss of a chromosome or by gain of the chromosome. What is the reason for this, or what is the cause, how it is caused? This is due to the non-segregation of chromatids at the time of cell division. Clear? So the normal case, it should be 2n, but it deviated from the normal. There is a gain of a chromosome or lacking of a chromosome. So it is represented as 2n plus 1, then it is called trisomy. 2n minus 1, it is called monosomy. So here remember, it is the change in the chromosome number. Now talking about polyploidy, is the change in the number of sets of chromosomes. So remember the difference. Here is the number of chromosomes, here is the number of sets of chromosomes. We know an individual has 2 sets of chromosomes, 2n. So this is normal, but in polyploidy it is abnormal. It is not 2n, it becomes 3n or 4n or 5n. The reason is failure of cytokinesis immediately after the telophase. So you must know the reason for both aneuploidy and polyploidy. For polyploidy, the reason is failure of cytokinesis immediately after cell division. Actually, we know after karyokinesis it should be cytokinesis, but sometimes cytokinesis does not follow, and as a result the number of chromosomes increases in sets. It can be 3n, 4n, 5n, etc. So failure of cytokinesis after telophase. So of all these types of chromosomes, the numerical mutation is the most important. It can be asked as difference between aneuploidy and polyploidy. So once again reiterating the difference between aneuploidy and polyploidy. Aneuploidy is the change in the number of chromosomes, whereas polyploidy is the change in the number of sets of chromosomes. Second difference, aneuploidy is caused due to the failure of segregation of chromatids at the time of cell division, whereas polyploidy is due to the failure of cytokinesis after telophase. So both there are failures occurring. Then aneuploidy, there are 2 types. 2n plus 1, it is called trisomy, or 2n minus 1, it is called monosomy. So in 2n minus 1, one chromosome is lacking from the normal, whereas 2n plus 1, one chromosome is in excess to the normal. Because it's in excess, one chromosome is in 3 copies. That's why it's getting the name trisomy. There, all the chromosomes should be in double copies, but there we can see one chromosome is in single copy. Hence, it gets the name monosomy. So I'm reiterating the fact that aneuploidy, differentiate between aneuploidy and polyploidy is very, very important. So first you say the condition, and second you give the cause. What causes these? Aneuploidy and polyploidy. So once again I am winding up by saying aneuploidy is change in the number of chromosomes, either due to a gain of a chromosome or loss of a chromosome, and hence there is 2 types, trisomy, monosomy. Second, it is caused due to the failure segregation of chromatids at the time of cell division. Now talk about polyploidy. It is the change in the number of sets of chromosomes. Usually it should be 2n for an individual, but here it becomes 3n, 4n, 5n. And the cause is due to the failure of cytokinesis immediately after telophase. We know telophase is the last part of, or the last phase of karyokinesis. It should be immediately followed by cytokinesis. But what happens? It's not followed by cytokinesis. In that case, it will result in 3n. Again, if it's not followed by cytokinesis, 4n, 5n, 6n, like that it goes on. Clear? So, these are the situations of we can, by artificial selection, we can create vegetables which are 3N, 4N, 5N. It is not naturally occurring, it is induced, whereas 2N aneuploidy can occur naturally under certain circumstances, that is failure in conceiving or late conceiving, like Down syndrome, Turner syndrome, Klinefelter syndrome, where the gametes is a abnormality in gamete formation. So, this aneuploidy naturally occurring, whereas polyploidy is induced, and that in plants it is being induced for the benefit of man. And you all have learnt in 10th standard about from the wild cabbage you get different types of plants like the kohlrabi, kale, red cabbage, cauliflower, broccoli, etc. That is a result of mutation which is creating polyploidy in the descendants from the ancestor of the wild cabbage. That is the wild cabbage. I hope you have understood mutations. Thank you.